So we've discussed throughout the semester all the various macronutrients, including carbohydrates, proteins, and lipids. And now for the remainder of the class, we're going to be focusing more on how vitamins and minerals are impacting the metabolism of those carbohydrates and proteins and lipids. Since we just finished with our lipids, it made sense for this lecture, we will be discussing lipid peroxidation and antioxidant nutrients. So these are all the vitamins and the minerals associated with or having antioxidant properties. So although there are many components within our cells that can be oxidized and affected by reactive oxygen species, our lipids are the most susceptible, and that's basically because of those double bonds. So when we have our lipids undergo oxidation, that's called peroxidation, which is the auto-oxidation of our lipids when they're exposed to oxygen. And this is what is causing the rancidity of our fats. So usually fat goes bad because of this auto-oxidation or peroxidation that's occurring. It's not because the microbes have degraded it and made it no longer safe, or that there's a lot of microbes growing within it that you would encounter with carbohydrates and proteins. So our rancidity or our food going bad, the reason why we don't want to eat off-flavored fat is because that's going to have a lot of reactive oxygen species. Peroxidation is also the cause of many cancers. It also is associated with inflammatory diseases and also what is causing those nasty aging symptoms. So your wrinkles, etc. A lot of that has to do with the peroxidation. The reason why we want to limit the peroxidation that occurs is because it's going to further peroxidation. So if you have one lipid that becomes auto-oxidized, it's going to cause other lipids to become oxidized. That's also forming our reactive oxygen species, or our ROS. We also have superoxide ions and our hydrogen peroxide. And as I mentioned before, our polyunsaturated fatty acids are more susceptible than our saturated fats to peroxidation, and that's because of the double bonds, the multiple double bonds that those unsaturated fats have. So there are three phases to lipid peroxidation. Our first phase is initiation, and this is where our fatty acid is interacting with a metal. This is usually going to be iron is one of the more common ones, or an existing free radical. And then we have our second step, which is propagation, where our free radicals react with oxygen, producing more free radicals. So we're propagating the amount of free radicals or reactive oxygen species. And then our third and final step is our termination, where our peroxyl radicals transform into non-radical compounds. So we get hydrocarbons, aldehydes, alcohols, ketones, and some of these compounds are harmful and can be carcinogenic, or also contributing to the increase of risk of cancer. They can cause an immune response, so that's going to instigate the critical, constant, or chronic inflammatory state. So what is initiating the free radical formation? And we have many different environmental factors that are contributing to the formation of these free radicals. The most common ones that we have learned all throughout elementary school, etc., is radiation and pollution. So these, we all know that radiation and pollution are bad. Radiation, this is why people don't want nuclear power plants around them. Pollution, all the many reasons why smoking has been banned, and we're trying to decrease the amount of emissions in cars, is to reduce the amount of pollution, and therefore we are healthier because we have less free radical formation. Now, the cells themselves cause free radicals. If you think of the electron transport chain in our mitochondria, that is basically a bunch of oxidation reactions that are occurring. We're transferring electrons all throughout that mitochondrial matrix membrane, and that's how we're forming our ATP. As our mitochondria get older and more worn out, they become more leaky, and we'll have some of these free radicals that are able to leach out, and that can cause some peroxidation. In addition, this is the way that we instigate an inflammatory response. So our immune cells, especially part of our innate immune cells, they are actually forming these free radicals within their peroxisomes, and that is what they are utilizing as their first defense against bacteria, viruses, and parasites. So just as it causes damage to us, it can cause damage to other microorganisms. So that's how we fight any pathogens that are attacking us. So as you can assume, there's going to be some of these after you have an immune response, you're over the flu, we're going to have some free radicals that are still within the system that need to be cleaned up, and that can also contribute to lipid peroxidation. That's why we try to limit the amount of inflammation that we have when we're fighting various diseases. So how do we combat this lipid peroxidation? Well, we utilize antioxidants. So what is an antioxidant? Well, it is a compound that is inhibiting oxidation, and we have both natural and synthetic antioxidants. The ones that you're probably most familiar with is take more vitamin E, your vitamin C. If you're sick, you want to take more vitamin C. We also have selenium, our mineral. And then there are others that have some antioxidant properties, include our carotenoids and our polyphenols. Some individuals argue that these are not actual real antioxidants because, in contrast to our vitamin E, C, and selenium, when they act as antioxidants, they are resynthesized. Carotenoids and polyphenols, once they are oxidized and quench that free radical, they are no longer active. So some individuals will actually say that carotenoids and polyphenols are not, but for all intents and purposes, we will say for this class that they are. We also have a lot of synthetic antioxidants. We have BHA, BHT, and ethoxyquin. So there's also some controversy with some of these. Some individuals want them out of the food chain, and some individuals will also say that their children are sensitive to BHA and BHT, similar to red dye 40, and it causes mood disorders, etc., in children. But all of these are used as antioxidants in our food. What are some of the main properties of our antioxidants? Most have a phenolic compound. So our phenolic group has an antioxidant property, and they are working because they are reducing initiation or they interfere with propagation. So that's how our antioxidants are working. So how do they actually work? Well, our antioxidant, as denoted in the picture on the right-hand side, you can see we have this very generic antioxidant, and it donates an electron to our free radical. And so this is going to stabilize the reactive oxygen species, because this has an unpaired electron that's unstable, and so our antioxidant is donating an electron that is able to stabilize that. So by contributing these electrons, it causes the free radicals to be more reduced and less likely to damage the cell. Now, as this is a nutrition class, we are going to focus on discussing the nutrients that have antioxidant properties. One of the most common ones that everybody is most familiar with is vitamin E. So vitamin E was actually discovered in 1920. A lot of our vitamins were really unknown until the early to mid 1900s. A lot of them were discovered from the University of Wisconsin in Madison. So it was shown that vitamin E reduces fetal resorption in rats. So initially it was called the fertility vitamin. And its only metabolic function is as a cellular antioxidant. So it is stabilizing our cell membranes and saving if any reactive oxygen species come in contact with the cell, vitamin E is there within the cell membrane, and it can stop the initiation and propagation. Our generic name for vitamin E is, we have tocopherol and tocotriols. And in addition, we have various isomers of vitamin E, just like basically everything else that we have discussed. So all of our natural isomers are going to have the abbreviation of as a R. Here, back, if you read old papers, they may refer to it as D-alpha D tocopherol, so that is the old terminology. And then we also can synthesize vitamin E, so our synthetic isomers are referred to as S. So we have the old terminology is DL. So you don't have to know that for the exam, but this is just if you happen to be reading any papers related to the vitamin E, if you come across D or DL, this way you know if it's a natural or synthetic. In addition to the stereoisomers, we have various homologs. So we have alpha, beta, gamma, and delta. So these homologs differ in the location and the number of methyl groups within the chromomain ring. And this also changes the biological activity that the vitamin E has. So as I mentioned before, that vitamin E has various isomers and homologs. So we also have different categories. So we have tocopherol and tocotrienols. And this is why, because of all these different compounds that have vitamin E activity, why when you look at your requirement or your RDA for vitamin E, it is stated as an international unit. And that's because it's the standardization to account for the various activity levels of these different compounds that have vitamin E activity. The most common vitamin E are under the class of tocopherol. So here you can see on the bottom our tocopherol versus our tocotrienols. You can see the main difference here is the double bonds that the tocotrienols have compared to our tocopherols that do not have those double bonds. And you by no means have to memorize these structures. This is just here for your general information. And as I mentioned before, we have these different homologs. So we have alpha. This is the one, alpha tocopherol is the most active out of all of the vitamin E's. Next, we have beta tocopherol, and that's 40%, as 40% of the activity of alpha. And our gamma tocopherol is 10% activity of our alpha tocopherol. And this is the majority of the vitamin E that we're going to find in plant seeds, is in the gamma form. And then we have delta, and delta has about 1% activity of alpha, and this is the highest amount in Western diets. Compare that to the tocotrienols, which these, as a class, have only 25% the activity as our tocopherols. They are also less abundant in the human diet because they are high in palm oil, rice bran, and barley, and we just don't consume significant amounts of those within the human diet. They have high antioxidant properties, and they are also anti-carcinogenic. So, but all in all, our tocopherols are more active than our tocotrienols. And here, if you want to see the differences, we have the different R groups. So for our alpha, we're going to have R prime is going to be a methyl group up here. So here's our R prime. And for our R double prime, it's going to be another methyl group. Compared with our beta, we're going to have a methyl on R prime and a hydrogen. Our gamma is going to be flip-flop with a hydrogen and a methyl group, and our delta is going to have 2 hydrogens. You by no means have to know this, so please do not memorize. There are too many other important things that you need to know, and this is what Google is for. So if you really want to know it, you can look it up in Google at a later point in time. So because we have these different activity levels of our various homologs and isomers of our vitamin E, we need our normalization. So we need to know, well, if I am consuming RRR alpha tocopherol, how much of that do I need to meet my requirement? So we have these unit mass equivalencies for 1 milligram. So this is basically what our supplement companies are utilizing to determine if you've met your vitamin E requirements or how much they are putting in the multivitamin that you take. So our standard is our all-rack alpha tocopherol acetate. So we conjugate tocopherol to acetate because it stabilizes it, and it makes it to where it is not, by having it conjugated to the acetate, it's not going to be oxidized before it's consumed. So that acetate is stabilizing the tocopherol. So this all-rack is the same as our DL, so our old terminology. So that is saying that this is synthetic alpha tocopherol, and we have not determined. So all-rack is indicating that it can be a various mixture of R's and S's. So we haven't cleared that all out, so we have all-rack alpha tocopherol. So this is our synthetic. This was also based off of, this became our standard because of the ability of the rat to utilize it. Now, is that the best method? Does that make it the best standard? So we looked at fetal rat absorption, and like I said, that may not be the best standard to utilize. Our other one is our free all-rack, and that has a international unit of, or equivalency of 1.1. So every milligram of free all-rack, we have 1.1. So free meaning it is not conjugated to acetate. And then we have our RRR alpha tocopherol. So remember, this is our natural. So it could also be referred to as D-alpha tocopherol. And this has the highest. So one of the things that I'd like you to know, I don't want you to be able to spout off all these numbers out at me, but I want you to know that RRR alpha tocopherol has the highest equivalency, or the highest value, is the most active out of all these. And then if we conjugate that to acetate, we reduce that to 1.36. So you should be able to see that our free has a higher activity level. However, it's more stable and has a longer shelf life if we conjugate it to acetate. And so as noted here, our acetate is added, it's esterified to our tocopherol to stabilize it, and it stabilizes it because our vitamin D is only active after the acetate is hydrolyzed off of it, and that's going to occur within the small intestine. So here we have a diagram showing of how vitamin D is actually protecting the cell. So here we have our free radicals. They are affecting our membrane polyunsaturated fatty acids. We're going to get some reactive oxygen species. And remember, our vitamin E is going to be within the cell membrane. It's hanging out with the rest of the fatty acids. So it is going to encounter any of these free radicals. So it's going to stop the initiation and propagation of the peroxidation that's occurring. So we have our tocopherol or tocotrienol. It takes up the free radicals. And then you can see, as I mentioned before, with our natural antioxidants or our vitamins and minerals that act as antioxidants, they are able to resynthesize compared to what our carotenoids and polyphenols are able to. So here we can see our vitamin E takes on the free radical, and it actually passes it on to vitamin C. So our vitamin C is ascorbate, and it becomes dihydroascorbate, and then we have it go into our thiol cycle, and basically we're able to, we consume an NADPH and form an NADP, and that's how we are protecting our cells from these free radicals. So our vitamin E donates phenolic hydrogen to the free radical. It forms tocopheroxyl free radical that's converted to tocopheryl oxyquinone, and that's reduced back to tocopherol by ascorbic acid. So the biggest thing here that I want you to take out is that our vitamin E can be recycled, and it is passing on that free radical onto vitamin C, and we end up consuming an NADPH at the end to quench and prevent any further peroxidation. So in humans, vitamin E deficiency is very rare. Usually you end up seeing it if there is fat malabsorption. So if you have some issue to where you are not able to absorb fat, if you've taken some weird, I know there have been some fad diet things where it binds up all the fat. If you ever had those Wow chips that I think Lay's potato had back in the late 90s. Those can cause some fat malabsorption, and there are various other metabolic diseases that end up resulting in malabsorption of fat. But if we induce it, and it's unethical to induce it in humans, but we see with fat malabsorption, we can get cystic fibrosis, premature infancy. We have hemolysis of our red blood cells, neuromuscular dysfunction, cystic breast disease. So those are some of the deficiency symptoms in humans. But like I said, very uncommon, and usually it is more of a secondary effect due to a fat malabsorption problem. We do see vitamin E deficiency in ruminants. When we have vitamin E deficiency in ruminants, it is usually in conjunction with selenium. So selenium is one of those minerals that has antioxidant properties, and this is because it's part of the glutathione peroxidase, which we'll talk about in a few minutes. So in ruminants, we get white muscle disease in sheep, and it's more common than in cattle. So it's more prevalent in sheep, usually due to a deficiency in selenium, and depending on where you live in the country and the soil selenium, you can have low levels of selenium, and it's exacerbated by inadequate vitamin E intake. And basically, the muscles are very weak, they're very stiff, we have deterioration of the muscles, including the cardiac muscles, so we have basically heart failure that's starting to occur in these animals. So here we have a picture of a lamb that has white muscle disease, and you can see it has this stiff humped back, and so it really basically hurts them to walk. And if you look at the muscles upon doing necropsy, it's called white muscle disease because the muscles are very white. Now, poultry, we've been able to induce vitamin E deficiency, and when we've induced it, we get exudative diathesis, we get edema, which includes edema of the breast muscle, blackening of various affecting parts, and oozing of interstitial fluids from the body. And in addition, you can also get encephalomalacia, which is also referred to as crazy chick disease or cherry brain. So this you can usually detect in chicks between 2 and 6 weeks of age, and basically they are acting crazy because they have hemorrhages within the cerebellum, and the amount of vitamin E that it requires is very much dependent on the amount of linoleic acid. As you remember, our polyunsaturated fats is what's determining how much of our vitamin E that we require. So if we have a lot of polyunsaturated fats, we're going to have a higher vitamin E requirement. So here's a picture of a chick that has crazy chick disease. So basically it's having a hard time keeping its head up and with balance. So it looks like it has its head turned around. So this is what encephalomalacia looks like in chicks. And as I mentioned before, it's also referred to as cherry brain because you have this hemorrhage within the brain and the cerebellum. So here you can see the hemorrhage. So this bottom bird is the one that was vitamin E deficient, and you can see there's a lot more blood clotting within the brain, and that's because of the deficiency in vitamin E. And here we have an example of exudative diathesis, which basically we have fluids just oozing out of the skin. and the feathers, so they just really have unable to keep the fluids, and it makes all their vessels more leaky because you have so much peroxidation. We're not able to keep those nice tight junctions. So this is a pretty severe case of vitamin E deficiency. So cats are also susceptible to vitamin E deficiency. They get yellow fat, and this is basically observed more if you have a cat owner that really thinks cats need to only eat fish. As you recall, fish have a lot of polyunsaturated fatty acids. We have a lot of EPA and DHA, and those specifically have a lot more unsaturated or double bonds within them. So if you don't have adequate antioxidants such as vitamin E, you're going to get this yellow fat. So symptoms of this include anorexia, pyrexia, which is a big old fancy word for fever. And the animal is usually, the cat is pretty tender, and it has lumpy abdominal fat. So they just, they don't want to be petted. When you do, you can feel their abdominal, they're going to have lumpy fat. So next we're going to talk about vitamin C. Vitamin C is the water-soluble antioxidant. So vitamin C is water-soluble. I think I failed to mention that our vitamin E is fat-soluble, so vitamin C is water-soluble. Another name for vitamin C is ascorbic acid. Now, some animals can synthesize it. So humans, unfortunately, cannot synthesize it, but mice, rats, pigs, for instance, poultry, all can synthesize vitamin C. So when we synthesize it, when those animals synthesize it, it is coming from glucose, and it readily undergoes oxidation and reduction. As I mentioned, some animals can synthesize it, so this is going to be the, we have pigs, poultry, rodents. So basically, getting back to our very first lecture, or that first unit of lectures, you wouldn't want to use a mouse or a rat as a model for evaluating vitamin C metabolism in humans, because they can synthesize it. Now, a better example would be a guinea pig, for instance, or a hamster. They are unable to synthesize vitamin C, so they're a better model. Now, it's very much required for collagen synthesis. So collagen is that connective tissue. It is necessary for the formation of the hydroxyproline and hydroxylysine, and our vitamin C also enhances our iron absorption. So it has reducing properties, which is able to enhance our iron absorption. So what is the function of our vitamin C? Well, we already got a preview of it when we were discussing our vitamin E function, but our vitamin C is actually found in its highest concentration in the pituitary gland and the adrenal gland, and it's also in high concentrations around healing wounds. So why would we do that? And that's because we need it for that collagen. All of our scar tissue is made up of collagen, so it's going to be higher concentrations around those areas. We have some of the functions. It's mainly an antioxidant. So you could see vitamin C, in contrast to vitamin E, vitamin E was only an antioxidant. It really didn't have any other function, but our vitamin C has many more functions outside of its antioxidant properties. So it's an antioxidant. It prevents oxidation of other substances by free radicals. The most common free radicals, superoxide, hydroxyl radical, and our hydrogen peroxide. So this is one of its main functions, what we think about. In addition, we have our collagen synthesis, why we have it high around wounds. It's also a cofactor for catecholamines. And it's also a cofactor in the synthesis of carnitine. So what do we need carnitine for? And good example, if you can think of it, reminder, basically we need this carnitine to transport fat, transport our fatty acids into the mitochondrial matrix. So if you remember, our fatty acid CoA is replaced. The CoA is replaced with carnitine, and it uses carnitine to get through that inner membrane space in the mitochondria. Beta oxidation is occurring within the matrix, so this is needed to transport our fats into the mitochondrial matrix. So our bioavailability is actually pretty high. It ranges between 80 and 90%, regardless of the source. It's absorbed via sodium-dependent active transport. And the site of absorption varies with species. For instance, with the guinea pig, it occurs in the duodenum and jejunum, whereas the rat, it occurs in the ileum. And the majority of it is transported unbound. So it's not bound, it does not have a carrier protein, it is not bound to albumin. You'll notice later on, one of the common themes with a lot of our B vitamins is they utilize albumin to transport them, and a lot of our fat-soluble vitamins have a carrier protein that moves them around in the blood, but our vitamin C is transported unbound as ascorbic acid. So as I mentioned before, that it's important component of collagen synthesis, and that's because we need vitamin C. It's a cofactor, meaning that it has to work with the enzyme to have the reaction occur. So here we have proline, which is our amino acid. Within our collagen, it actually is converted to hydroxyproline. So we need hydroxylase, vitamin C, and iron. When we have vitamin C and iron acting as cofactors for hydroxylase, we add a hydroxyl group and we form hydroxyproline. So therefore, this is why it's without this, we're not going to be able to form our collagen, and we get some difficulty in wound healing if we have inadequate vitamin C. So here's a list of our various deficiency signs in animals and humans. So the deficiency really depends on which species you are referring to. So for instance, humans cannot synthesize vitamin C, and the main deficiency, as you may have all heard of, is scurvy. And this was very common in sailors. They used to develop scurvy all the time until it was discovered, well, they needed to have fruit on their journeys, and all fruits are high in vitamin C. Specifically, our citrus fruit has a lot of vitamin C or ascorbic acid in it. But our scurvy, what are some of the signs? We get the atrophy of the gums. We have bone and cartilage formation stops. we got a lot of destruction of our red blood cells. We have endocrine problems, and eventually we will have multiple organ failure. So we can also see various symptoms similar to scurvy in fish and our non-human primates and guinea pigs. Our ruminants, they can synthesize vitamin C. They still need some in the diet. And they typically, if you're going to provide it in the diet, they need more than a non-ruminant because the vitamin C is destroyed in the rumen due to the microbes. Our swine and poultry, for the most part, they have adequate amounts. We do provide some in the diets of the young poultry and swine, and that's because of the rate of growth that they are enduring. So by providing that, they just cannot synthesize enough vitamin C necessary for their the rate of growth that they've been genetically selected for. Dogs and cats, they can synthesize it. And as I mentioned, fish, non-human primates, and guinea pigs cannot. So you'll see more of the symptoms within our similar to scurvy in all the animals that cannot synthesize vitamin C. So here's a diagram of an individual, a sketch of scurvy. So a lot of these individuals, they lose their teeth, and a lot of that is because since you're unable to synthesize collagen, you end up getting a lot of your gums cannot maintain themselves, so you lose the integrity of your gums, so therefore you'll lose your teeth. Also due to the fact of the effects on the bone and cartilage, usually their pale skin, very sunken eyes are some of the signs of scurvy. So here we have the periodontal disease associated with scurvy. So as I mentioned before, our gums are affected. So we have receding gums due to the lack of resynthesis of collagen, and that ends up exposing the roots of our teeth, and then bacteria can grow in there, and that's what is going to end up causing infection and dental caries, and eventually you'll lose your teeth if it goes on for too long. So toxicity with vitamin C in humans, it's generally, and animals, it's not really thought to have any toxic effects. Because it is water soluble, you can excrete a significant amount. You can take up to 10 grams per day, but I could tell you that any levels greater than 2 grams per day, there's no added benefit. The max your body will absorb is 2 grams per day. It has been associated with enhanced absorption of iron. So therefore, if you are, we could exacerbate some reactive oxygen components, so we're increasing our iron absorption. If you have adequate iron already, then if we have too much vitamin C, it could actually cause some peroxidation. And it doesn't seem to increase oxalate excretion in kidneys, and it does not make the body pro-oxidative. As I mentioned before, animals, vitamin C toxicity does not seem to be a problem. So we really don't have to worry about toxicity of vitamin C at all. Now, some people like to take vitamin C if they feel a cold coming on, so there have been some studies that have shown that you can decrease the duration of the common cold if you take supplemental vitamin C. I believe the product Airborne has higher levels of vitamin C within it. that people take in hopes to reduce the length of their cold. And in addition, higher levels of vitamin C is helpful for elderly people who are less able to absorb the vitamins. So this basically goes along with all of our nutrients. The older you get, the less efficient you are at absorbing your nutrients. So therefore, our elderly individuals should take higher levels, or they should have a higher RDA of our vitamin C and other vitamins within their diet. So our last antioxidant nutrient that we're going to talk about is selenium. So selenium works in conjunction with vitamin E. It helps prevent the propagation of fatty acid oxidation in membranes, and because of this, it's said to have a sparing effect. It is a component of glutathione peroxidase, and which is the second line of defense against free radicals. So glutathione peroxidase stops the propagation. And the difficulty with selenium is the concentration within the soil varies greatly in the U.S. from very deficient to extremely toxic. So therefore, especially in this global economy where we have, for instance, a lot of our grains grown in a specific area, some of those areas, one side of the state is toxic, the other side is deficient. So you have to verify the amount of selenium within your feed ingredients. And one of the reasons why it is toxic is it can replace the sulfur within cysteine and methionine. So if you've ever heard of your organic trace minerals, that's where, so our organic selenium is selenomethionine. So instead of sulfur in the methionine, it's going to have selenium. And that ends up, as you can recall, our cysteine and methionine are, our cysteine is used in disulfide bonds, so we're going to lose, if we have too much selenium in the form of selenial cysteine, then we are going to not be able to form our double bonds, and we're going to have improper folding. And then with our methionine is involved in our one-carbon metabolism, which we'll talk about later on in the semester. In addition, it's also involved in our disulfide bonds. So we're going to have misfolding of protein. We're also going to mess up our one-carbon metabolism. So basically, our selenium, the toxic effect is more due to when it's in this organic state. Our inorganic is less toxic than our organic selenium. So here we have that same reaction scheme that I showed with how vitamin E works with selenium. However, selenium is associated with the glutathione peroxidase. So here, as we discussed before, we have our free radicals affect our membrane polyunsaturated fatty acids, and our vitamin E is sitting within our cell membrane. It's able to take those and pass it on to ascorbic acid. That then forms dihydroascorbate. And then we utilize glutathione, and it's the enzyme glutathione peroxidase is occurring right here. So we have our glutathione peroxidase, which is then converting it to alpha lipolytic acid, or oxidized glutathione, known as GSSG, and then we utilize our NADPH again. So our glutathione peroxidase is occurring mainly within the cytosol. of the cell, and it's reducing the peroxide. So we have vitamin E is our first line of defense, preventing any external reactive oxygen species from getting into the cell, protecting the almighty DNA, because if we oxidize our DNA, we're going to be at a greater risk of cancer. And our glutathione peroxidase, or glutathione, is our secondary defense. So if these peroxides are able to, or reactive oxygen species are able to come through into the membrane, and we should be able to utilize our glutathione peroxidase and quench those reactions also. So when we have them with our vitamin E, we're utilizing vitamin C, and then our glutathione or thiol cycle. In addition, we also have some glutathione peroxidase occurring within the mitochondria. So if you recall, that's where our electron transport chain is, and where we're more susceptible to making endogenous reactive oxygen species. So as our mitochondria get older and less efficient, we're going to leak out. It's not 100% efficient closed system, so we're going to leak out some of that, those reactive oxygen species. So with that, instead of using the ascorbic acid here, we utilize the superoxide dismutase enzyme, which you may have heard. I believe Cindy Crawford sells a product that has higher levels of superoxide dismutase, which in theory should reduce the effects of aging on your skin. So our superoxide dismutase is able to take our reactive oxygen species, it forms a peroxide, and then our glutathione peroxidase reduces that, and we still get the formation of our NADP from our NADPH. So if it is coming, if the reactive oxygen species are coming from the mitochondria, the theory is that in that sense, our glutathione peroxidase is our first line of defense, because we're trying to limit that exposure of our cell membrane, because we want to contain that potential reactive oxygen species so it doesn't spread to other cells. So here I have a map of the United States showing the amounts of selenium within the soil. So you can see the blue areas, the dark blue throughout Oregon and Washington, and so the Northeast and the Northwest, you could see, and in Florida, you could see that there's a very low amount of selenium within the soil. And then we have various areas that we have variable amounts. So the majority of California, Nevada, Idaho, Utah, we're going to have, and then North Carolina, South Carolina, Georgia. So the South, East, and the California, Nevada region, you can see they have variable amounts. All the white components are adequate, and then our red areas is where we have toxic levels. So you can see that, especially through parts of the Dakotas and our Wyoming, that we can have in Nebraska, we have a lot of areas within the Great Plains regions that can be quite toxic, and we do have a significant amount of our grains being grown in this particular area. And it's because of this variable level of selenium within the soil that the federal government actually restricts the amount that selenium can be provided within an animal's diet, because we don't want the animals to have toxicity signs of selenium. So just as I mentioned before, with vitamin E, our deficiency sign in ruminants, same with selenium, is our white muscle disease, and that's due to deficiency in selenium, but it's exacerbated by the vitamin E intake. So if you have low selenium and low vitamin E, you're going to have a severe case of white muscle disease compared to if you had adequate vitamin E and deficient selenium. So our symptoms, again, weakness, stiffness, deterioration of muscles, including. So here we have another reminder of that sheep that has the white muscle disease. Again, you can see it's stiff, humpbacked, and that makes it more difficult for it to walk. And this is an abnormal posture for a lamb. So you could tell that this animal is having a difficult time walking, and it's also not growing like its counterparts. So that's where we have a good indication that this is vitamin E and selenium deficiency. So this is what the white muscle disease looks like. So you can see on the left-hand side the lamb that has white muscle disease. The muscle is very white. It's lost a lot of its pigment. Not saying that the red fibers have been replaced with white muscle fibers. It just does not have its pigment. The myofibril pigment is not there. And on the right-hand side, you can see our normal muscle tissue. So you can see it has that dark red pigment that you would anticipate, especially with lamb. So here you can see the white muscle disease, and it's referred to that because we have these white streaks within the hindquarters, actually everywhere. So because these muscles are not able to function correctly, that's why that lamb had difficult time standing and why it was hunched and looked like it was in a significant amount of pain as it walked. So here you can see the effects of white muscle disease on the heart. So as I mentioned before, it does affect cardiac muscle tissue also. So you lose the pigmentation in the heart, and basically the heart becomes so flaccid, and just like the animal was not able to walk very well, the heart eventually fails to beat, and that's what's going to cause the animal to die, is it dies of heart failure. And if we do histology on the heart muscle, you could see a significant amount of infiltration of our white blood cells occurring, and that's basically because especially our macrophages are trying to clean up the area. They're trying to prevent any disease that may be causing these cells from dying. So that's why we have a lot of infiltration of our white blood cells. So as I mentioned before, selenium is one of the more toxic minerals, and that's because of its ability to replace sulfur within cysteine and methionine. Rarely do we see selenium toxicity in people. Usually you're going to see that if people are taking extra supplements, or if they live in areas that have that toxic soil level. In cattle, you're going to see hair loss, lameness, and hoof problems. And all of that, if you remember your cysteine, so this a lot has to do with the sulfur amino acids. So we're replacing the sulfur with selenium, and therefore we lose proper folding of the protein. So that's going to make the hair brittle. Eventually, we're not going to be able to grow that back as it falls out. Lameness and hoof problems because we have compromised the integrity of the keratin protein, and that's what's going to cause them difficulty walking. Our poultry, if you have too much selenium, you're going to see decreased hatchability and increased embryo deformities. And horses, again, you're going to see cracked hoofs and loss of hair similar to our cattle. And once again, that's mainly due to the misfolding of the proteins because we've replaced the sulfur with selenium and are unable to form those disulfide bonds anymore. So here we can see indication of some selenium toxicity, and I will let you guess where this particular steer is being raised, which area where it would be subjected to selenium toxicity. And you can see that it is very lame in the front legs, and it has a very poor conditioning within the body. And here, as I mentioned before, you have deformed hooves, and a lot of that has to do with the fact that with this toxicity, we are replacing the cysteine and methionine with selenomethionine and selenocysteine, so therefore we lose the disulfide bonds and have misfolded keratin protein. So here you could see the effects within the hooves of these cattle, how we have these deformations. And like I said, that's all due to misformations of or misfolding of the keratin proteins. And here is our example in the horse. You could see we have a horizontal crack in the hoof. So that's definitely an indication. So here's our crack in the hoof. So we know that that's due to the growing. So the hoof growing, and it's starting to. We now have a significant amount of selenium within the diet for a prolonged period of time. We're now getting chronic effects. So our cracks are not going to be vertical, they're going to be horizontal. In addition, you can see the sparse hair on the tail over here. So the horses should have much longer hair on their tails. And again, this is all due to the organic form of selenium. If you're feeding an inorganic, you're going to have a lower incidence of the toxicity in an inorganic form. It's more with these organic forms that you have a higher rate of toxicity with selenium.